Hello everyone, in this video we'll discuss about G6PD deficiency. We will discuss in detail the role of G6PD enzyme and why does its deficiency cause hemolysis. Now G6PD deficiency is an X-linked disorder, and as the name suggests, there is reduced activity of G6PD enzyme in the red blood cells and there is a presence of hemolysis. Now, understanding why does it cause hemolysis? Firstly, we'll understand the role of G6PD enzyme. Now, in our body, oxidants are produced such as hydrogen peroxide, and these are normally converted into water with the help of reduced glutathione. Now, this reduced glutathione is produced with the help of an enzyme known as glutathione reductase, which requires NADPH for its activity. Now, this NADPH is produced from the help of G6PD enzyme. So, if this enzyme is deficient, this will lead to a no NADPH. Further, there will be no glutathione reductase enzyme, and oxidants like hydrogen peroxides, they will increase in the body. Now, understanding what would happen is when there is deficiency of G6PD, we already understood that there is decreased NADPH, then there is a decrease in the reduced form of glutathione, then there is accumulation of oxidants like hydrogen peroxide, and this will cause oxidant damage to the red blood cell. It will cause oxidant damage to the red blood cell membrane, which will make this red cell defective, and therefore it will go hemolysis in spleen, and therefore it will cause hemolytic anemia. Now, going to the main categorization. What is the categorization in G6PD enzyme? It depends upon the G6PD enzyme deficiency. So it can be characterized as class 1 if there is G6PD enzyme activity is less than 5% of the normal. So it will cause this form of category is very rare. It is severest form and it will cause congenital hemolytic anemia. Then if the enzyme activity is less than 10%, it will cause hemolysis. It will cause episodic acute hemolysis. It will be not self-limited, and oxidant drugs these can cause. Then there is category 3 in which there is 10 to 60% of normal enzyme activity. So it will cause self-limited hemolysis after the drug or infections. And lastly, there is category 4 and category 5. So we will understand this symptoms later on also. Now uh, going to the genetics part. Firstly, we already understood that this is a X-linked disorder. So as you know, mostly X-linked disorder, they occur exclusively in the females, because in females they occur exclusively in males, because in females there is lionization, because there is random inactivation of the X chromosome, and the second one is converted into bar body. So in females the expression is very variable. Now going to the clinical features. Now clinical features, firstly, it will depend upon the category. It can be asymptomatic. Patients can be asymptomatic, especially in category 3, category 4, category 5. They will be asymptomatic. But in category 1, 2, it can cause neonatal jaundice. In other categories, it can cause hemolytic anemia and exposure to oxidant stress. We will understand which are the oxidant stress, and it can also cause chronic hemolytic anemia also. Now going to the what is oxidant stress, and there are many causes which can be classified into oxidant stress for the RBCs. There can be infections like bacterial and viral infections. There can be drugs like anti-malarial drugs, mainly primaquine is very important for that, antibacterials like sulfonamides, then antipyretics and analgesics. Then there are many chemicals like naphthalene balls, methylene blue. Whenever the body is exposed to these chemicals, these act as a oxidant stress. They act as an oxidant stress condition, and these can further cause increase in hydrogen peroxide, which can cause increase in hemolysis, and then the symptoms will come. Okay, so the symptoms, they can vary according to the class. Example, in class 3, the hemolysis will be mild to moderate and will be self-restricted. That means the hemolysis will stop it is self-limited, and it will stop even if the patient takes that offending drug, because only the older cells will be killed in this category. However, in class 2, hemolysis will be marked and non-self-limited, because this is a type of enzyme deficiency in which not only the older red cells they are deficient, but younger red cells are also deficient. So here the hemolysis will not be self-limited. So symptoms will totally different from the class to class. Now going to the lab investigations. Firstly, on blood smear. This is the primary investigation. So on the blood smear, what you will see is there will be in this picture you can see these are the bite cells. You can see these are the bite cells. So what is bite cell? Whenever the red cell membrane is damaged, what happens is the splenic macrophages, they take a bite of it. They remove the dirty part, the damaged part of the red blood cell. This is known as the bite cell, and this is known as the blister cell. This one, in which or the ghost cell, in which a half of the RBC contains hemoglobin and other part is just empty. So these are the blister cell or the ghost cell. And there is another thing known as Heinz body. Firstly, there is also polychromasia in the red cell morphology. That means there will be different colors which of RBCs you can see. So you can see this RBC has a purplish hue to it. Then there will be some RBCs which will be pink in color. So there will be polychromasia, and then there will be presence of also spherocytic cells. There will be numerous spherocytic cells which will be seen. Uh, then again, there is a one thing known as Heinz body. Now, what is Heinz body? Heinz body, these are the red blood cell inclusions which are attached to the RBC membrane. You can see the blue dot over here. So these are only seen on the supravital staining such as methylene blue, methyl violet. So what is it is? It is the denatured hemoglobin which is present at the corner. So because of the oxidant stress, the hemoglobin gets denatured and it is present over here. Heinz bodies, they are very important in G6PD deficiency. In biochemical investigation, we will not go in detail because this is characteristic of intravascular hemolysis. There will be increase in unconjugated hyperbilirubinemia, there will be decrease in haptoglobin, there will be hemoglobinemia, hemoglobinuria. I will discuss in separate why the features which are different in extravascular hemolysis and intravascular hemolysis. Now there are some tests which are used for detection and screening of G6PD deficiency, especially in endemic areas. So we are not discussing it in detail, but the names are: firstly there is fluorescent spot test, there is myth hemoglobin reduction test, there is diet decolorization test, and there is some quantitative tests done for G6PD deficiency. Now what is the fluorescent spot test? Just briefing it. So whenever the G6PD is present in the blood sample, so that it's normal person, it causes a conversion of NADP to NADPH, that we understood. Now NADPH, it fluoresces in presence of the UV light. So in this test we use some reagents so that the fluorescence is seen. If fluorescence is not seen or is decreased, we can see that there is G6PD deficiency. So this was all about the G6PD deficiency. Do ask any queries regarding this topic in the description box. Also let me know the topics you want me to discuss. Thanks for watching this video, and like and subscribe to this channel if you like these types of videos. Thank you.